Xavier's College, Kolkata. The topic of the presentation is Synergistic treatment with curcumin and all-trans-retinoic acid. Effects on MMPs in metastatic breast cancer setting.
Very good afternoon to respected professors and all my fellow participants. I am Anirban from St. James College, Kolkata, from the Department of Biology, and I am working under Dr. Anirban Banerjee. And I am going to present my work, Synergistic treatment with curcumin and all-trans-retinoic acid, effects on matrix metalloproteinases, MMPs, in metastatic breast cancer. So we know, according to the research and experimental data that has been published by the World Health Organization, we know that around 11.7% of total cancer cases that are diagnosed globally, which accounts to 22,61,419 cases that of breast cancer. And in India, among females, we see around 178,361 cases, which is 26.3% of total cancer diagnosis that has happened. And we know that breast cancer is one of the most common heterogeneous disease which is affecting women in India and globally. And one of the key role factors in the process of treatment, we see that often in late diagnosis, the containing of cancer and treatment becomes quite difficult because of the process of metastatic spread of cancer from primary site to distant organs. And the key role players in the process of metastasis are matrix metalloproteinases or MMPs. The MMPs is independent endopeptide medicines family of proteins, and these MMPs, especially MMP2 and MMP9, that is gelatinase A and gelatinase B, they are very much has been reported in various experimental research in various forms of cancer. That these two MMPs play very important role in the degradation of extracellular matrix, especially the collagen 4 component of extracellular matrix, and help in the improvisation of the tumor cells from the primary site to the extracellular medium. And now these MMPs, they are mostly secreted as zymogens, so they need to be activated. So what happens, especially in the case of MMP2 and MMP9, is a gelatinous structure, and we see that there is a signal peptide followed by a propeptide domain, then we have the catalytic domain followed by the hemopexin domain. Hemopexin domain helps in substrate recognition, that is substrate binding, and the catalytic action happens at the catalytic site. But there is the pro-domain, it generates a cysteine switch, because of which what happens, the substrate cannot bind to the catalytic site. So in order to activate these MMP2 and MMP9, the cleavage of the pro-domain is very much necessary. And that is carried out by certain other proteins or MMPs. Like we have the MT1 MMP, that is the membrane-bound matrix metalloproteinase 1, which helps in the activation of MMP2. But in case of MMP9, we have other proteins like plasmin. Similarly, for MT1 MMPs, or which is also known as MMP14, they are also released as proenzymes or zymogens. So they also have to be activated. They are activated by other class of proteins known as furin, because they have a furin binding domain. Now, we know that there are various cellular signaling pathways, aberrant cellular signaling pathways, which gets downregulated in cancer. And among them, in our study, we have explored two important pathways. That is the integrin-mediated focal addition kinase pathway and the phosphatidylinositol 3' kinase pathway, or the PI3K pathway. Now, these two pathways have played a very important role. It has been already reported that it plays a very important role in the activation and in the aberrant expression and activity of matrix metalloproteinase 2 and matrix metalloproteinase 9, MMP2 and MMP9. Now, in the modern treatment of cancer, we know that, especially in the case of breast cancer, when it is often at a late diagnosis, often it takes an extensive period of treatment. Often it ranges from months to years sometimes. But the main problem with the use of chemotherapeutic drugs, of course, they are working a lot and they are used extensively in treatment nowadays. But the main important problem of using the chemotherapeutic drugs is the compromisation of the life expectancy of the patients, often because of extensive treatment. And often we see, like my father is also a patient, he is also a survivor from prostate cancer. And when I went to the hospital, I've seen that breast cancer patients, they have to undergo tremendous treatment. And some people even they die within the process of treatment. They cannot take the therapy of chemotherapeutic drugs. So because of that, we often see this development of this new arena of research that has come with the use of natural compounds from various plant and animal sources. And in our study, we have taken two important phytochemical compounds. What is the all-trans-retinoic acid ATRA, which is often from carotenoid-rich fruits and vegetables. And then we have curcumin, which is often from curcumaloma, that's the turmeric plant, rhizome of the turmeric plant. And curcumin has very important antioxidant and anti-inflammatory activities. In case of ATRA, we already know that it has already shown a very good effect in acute promyelocytic leukemia. The objective of the study includes to analyze the importance and effect of synergistic treatment with all-trans-retinoic acid and curcumin in downregulation of expression and activity in matrix metalloproteins 2, matrix metalloproteins 9, MT1, MMP, and on the signaling pathways regulating MMP in metastatic breast cancer cells. Now, in the methodology that we have applied, we have studied the interaction of ATRA and curcumin and synthetic inhibitors with MMP2, MMP9, MT1, MMP, focal addition kinase and phosphatidylate 3' kinase, that is PI3K, which were determined by computer-based techniques. Then we have we have treated the highly metastatic breast cancer cell line MTMB453 with ATRA and curcumin, both by single and as well as synergistic treatment. And the effects of treatment of MMP2, MMP9, MT1, MMP, focal addition kinase, PI3K, and the activity of MMP2 and MMP9 has also been analyzed post-treatment. Now, in the molecular docking analysis, we see that ATRA and curcumin has shown very good gelatinase A binding affinity with values of -10.11 and -10.99 kilocalories per mole, which was often after the analysis of the molecular docking complexes. But we have also studied with batimastat and doxycycline. These are two MMP inhibitors already used in treatment, and we see that they also show affinity for MMP2 catalytic domain, that is gelatinase A catalytic domain, that is with the values of -9.62 kilocalorie per mole and -6.64 kilocalorie per mole for batimastat and doxycycline respectively. And in comparison to batimastat and doxycycline with the negative values, with the lower values of -10.11 and -10.99 kilocalorie per mole, we see that ATRA and curcumin has shown a better affinity for MMP2 catalytic domain. Similarly, for MMP9 also, we see that ATRA and curcumin has shown a very good binding affinity for gelatinase D catalytic domain with values of -11.13 and -11.23 kilocalorie per mole. In comparison, we see that batimastat and doxycycline has shown values of -6.42 and -7.28 kilocalorie per mole of low or mean binding energy. That is calculated from the molecular docking complexes. Now, we have also studied with MT1 MMPs because they play very important role in activation of MMP2. So, MT1 MMP and ATRA curcumin interaction, we see that values of -8.28 and -10.78 kilocalorie per mole for ATRA and curcumin. We have shown a very good binding affinity for MMP14 catalytic domain because MT1 MMP is also known as MMP14. And similarly, like the previous experiments, we have also studied the interaction of batimastat and doxycycline with values of -5.45 and -7.12 kilocalorie per mole. We have treated the MGMB, that's MGMB453 highly metastatic breast cancer cell line, with singular treatments of curcumin and ATRA and with synergistic treatment of curcumin and ATRA, 20 micromole per ml of concentration. And we see that in comparison to singular treatments of ATRA and curcumin, synergistic treatment has shown a significant decrease in expression of MMP2 and MMP9, and as well as in case of MT1 MMPs post-treatment. And also we have analyzed the activity of MMP2 and MMP9 from two types of samples after treatment, that is cell lysis and SSTM samples. And we see that in comparison to the singular treatments of curcumin and ATRA, the synergistic treatment of curcumin and ATRA have decreased the activity of MMP2 and MMP9 in MGMB453 cell line. These two, sir, the lower one. So these are the activities. This is the zymogram. This is gelatin zymogram that we have done. This is the activity of MMP2 and that is the expression, and this is the activity. We have done gelatin zymogram. So from there, this is singular treatments, and this is the synergistic treatment. Now there are two types of samples. Now just because let me cultured the cells in the petri dish medium. We do it in serum-free culture media, that is the SFCM sample. So just because MMP is a secreted as well as cell bound, so we have taken cell lysis samples also. This is the extraction that we've done from the cell plate, and this is the SFCM, that is the medium. That is also collected, and from here also we have done the. Now, as we know that focal adhesion kinase, integrin-mediated focal adhesion kinase leads to the activation and over-aberrant expression of MMP2 and MMP9. So we have also studied the interaction of focal adhesion kinase with ATRA, with curcumin and with defactinib. Defactinib is a focal adhesion kinase inhibitor. And in comparison to defactinib, we see that the ATRA and curcumin has shown good binding affinity with focal adhesion kinase with -7.24 and -7.54 kilocalorie per mole. We have also treated the MTMB453 cell lines with singular treatments of curcumin and ATRA and with synergistic treatment, and we see that the synergistic treatment in comparison to the singular treatments has significantly decreased the expression level of focal adhesion kinase. We have also studied the interaction of PI3K, that is P110 alpha catalytic subunit, because PI3K is composed of two subunits, one is P85 regulatory subunit and one P110 alpha catalytic subunit. So we have studied with the catalytic subunit the interaction, and we see that ATRA has shown a considerable good binding affinity with -6.54 kilocalorie per mole. But interestingly, in case of curcumin, we have seen that it has shown not very good, but proper binding with -4.72 kilocalorie per mole. But when we take the alpelisib, which is also a synthetic inhibitor, we have seen that the values are -7.13 kilocalorie per mole. But when we have validated that in our wet lab experiment, we see that with treatment of MTMB453 cell line, curcumin and ATRA has considerably decreased the expression level of phospho-PI3K, and we see that the synergistic treatment has also decreased the expression of curcumin and ATRA in comparison to the singular treatment. So thus in a discussion, we would like to conclude that computer studies show high binding affinity of ATRA and curcumin to MMP2 and MMP9 and MT1 MMP in comparison to synthetic MMP inhibitors like bimostat and doxycycline. Singular and synergistic treatment of highly metastatic MTMB453 breast cancer cell lines with ATRA and curcumin significantly decreased in MMP2 and MMP9 and MT1 MMP expression. MMP2 and MMP9 activity in both cell extract samples and SFCM samples supernatant were also appreciably decreased. The molecular docking analysis showed that ATRA and curcumin showed high binding affinity for focal adhesion kinase and PI3K. PI3K comparable compared to synthetic inhibitors of defactinib and alpelisib. Singular and synergistic treatment of MTMB453 breast cancer cell lines with ATRA and curcumin significantly decreased FAC and PI3K. Synergistic treatment with both ATRA and curcumin appeared to be more effective in inhibiting MMP2, MMP9, MT1, MMP, FAC and PI3 in comparison to singular treatments. And as the MMP14 plays a very important role, as mentioned before, also in MMP2 activation, JAK regulation could lead to lower MMP2 activity. Inhibition of MMP2 and MMP9 expression and activity and MT1 MMP expression could lower breast cancer metastasis, tumor progression, secondary tumor formation. So I would like to thank my guide, Dr. Anirban Banerjee, for his continuous guidance and support. And I would also like to thank our principal, Father Dominic Savio, for providing his research facilities and Central Research Facility for providing all the facilities for my doctoral study. Thank you.